and there is no recessive. They are codominantly expressed. And here the leukocyte, and here the MHC, which is encoded on the cell surface of the leukocyte, and here the set from the mother, and here the set from the father. And for example, here the father mate with the mother, and they have children, and this is the HLA of the father, and this is the HLA of the mother, and the HLA of the 25% of the children, A1, 51, and 2, 7, and this is the haplotype. We read it horizontally, AB, AB, 1, 51, 2, 7. This is from the mother, this haplotype from the mother, and this haplotype from the father. And this constitutes 25% of the offspring. And the another 25% of their children have the another haplotype, 28, 27 from the mother, and 33, 18 from the father. And the another 25% of their children, their HLA typing is 1, 27, and 2, 18. And the rest, 25% of their children, 28, 51, and 33, 7. This chiasmatically, the inheritance is called crossover genetic recombination. There is a chiasmatically inheritance, which is rare. So this follows the Mendalian laws of genetic, which is 25% of siblings share 2 haplotypes, 50% of siblings share 1 haplotype, and 25% of siblings share no haplotype. This is Mendalian law. Thus, 2 random individuals are unlikely to have identical set of HLA molecules. So each one of us, HLA typing of him is not similar to another person. So reject organ transplantation and differ in their susceptibility to disease. Now we have a new subject, HLA nomenclature. How can nominate the HLA typing? According to method, we did the test serological nomenclature, which is the HLA, and this is the locus, and this is the allele. While in the molecular nomenclature using DNA typing, this is the locus, and we put a star. When we see a star, so once we see a star, so we can understand that this HLA typing done by molecular method, and here the allele group, and here the allele number, and this is the allele group, which is similar to serological method, and here the allele number, and here the silent polymorphism in exon, and also give it a number. And this is gene coding for beta chain. This is for beta chain. When there is A, so for the alpha chain. Genetic polymorphism. Why there is large number of HLA alleles? And this is due to genetic recombination, crossover, as here. One segment translocated to the another segment. So this leads to production of new allele. Intra-allelic conversion, and here the rotation of the chain of the DNA, and gene conversion. The gene, there is a conversion in it. And last one, the mutation. All these factors lead to large number of HLA alleles. Now the new subject is tissue typing. What's tissue typing? Is a laboratory test used to determine the type or the number of alleles to each person in each locus. HLA typing done by following methods: serological method and molecular method. And molecular method of two types, low resolution and high resolution. Serological method done first by scientist Terasaki and McClellan in 1964, while molecular method genotype level uh, of many types, SSP, which is sequence-specific primer, and another method, RASCA, which is reference transconformational analysis, and the newest one, which is NGS, next generation sequencing, and DNA sequencing. Sometimes you use the letter W, which is mean workshop, and this is rarely used because of the technique, molecular technique. And this is mean that the allele is not well recognized and still under study. So thanks for NGS that leads to change in analysis HLI locus due to its high accuracy. Uses of HLI typing. First, paternity testing in forensic medicine to prove the identity of illegal baby. And disease association, most common disease, Bahjat disease with HLA-B5 and ankylosing spondylitis with HLA-B27. Organ transplantation, which is very, very important, kidney, liver, heart, lung, etc. Control immune response by MHC restriction. Class 1 present antigen to cytotoxic T cell, while the class 2 present antigen to T helper T cell. Anthropology, study race and nation, because Caucasian is different from African, different from Americans. So the race, each race has its own HLA typing. Causes of MHC. And why there is what is the control of the HLA alleles production? First, transcription factor, class 2 transcription factor. This is class 2 transcription A factor. Regulatory factor X, regulatory factor X. Defect in these two factors leads to bare lymphocyte syndrome. Second, cytokine, which is alpha interferon, beta, gamma, tumor necrosis factor, interleukin 4. Gamma interferon increase the class 2 expression. Drugs, corticosteroid and prostaglandins decrease expression of class 2 molecules. So use cortisone in high dose lead to decrease in immunity, and this is due to decrease expression of class 2 molecules. Causes of MHC and disease stability. There are a number of different diseases associated with a particular HLA alleles. As we said previously, HLA-B27 and ankylosing spondylitis, and B5 with Behçet disease. And why these associations occur between the HLA alleles and these diseases? First, molecular mimicry between HLA antigen and given pathogen. So the immune system, instead of attack the pathogen, will attack the HLA antigen. Second, immunologic cross-reaction in infected individuals. Third, contribution of linkage disequilibrium, linkage between HLA ABC with the RDQ, close linkage of tumor necrosis gene with HLA-B locus, tumor necrosis factor gene, which is present in the class 3, which is close to the class 2. MHC, another reason, MHC genes encode molecules that serve as receptors for the pathogen. There are the second part, which is the non-classical. We said previously all the classical part of the class 1. Now we have the non-classical class 1 type, which is include 4 locus, which is recently discovered, E, F, G, H. Their functions were unknown except HLA-G, and these are also polymorphic, and with time they increase in its numbers, like the classical type. And the HLA-G expressed on the cytotrophoblast at the fetal-maternal interface that protect fetus from being recognized as foreign and leads to abortion. And the another function, HLA-G exerts tolerogenic function involved in the transplant acceptance. And lastly, HLA-G was important in tumor and viral immune escape. And this is the most function of the HLA-G. And thank you.